All right, hello everybody. This is Professor Howard for the second round of Chapter 6 screencasting. The other PowerPoint, I don't know what happened. It must have gotten corrupted or something on my computer. It was just a different, way shorter, weirder version than this one. So I thought I'd upload both just because the other one had some useful information about tumor suppressor genes and cancer. But this one actually has the meat of mitosis. So because I talked about some of this stuff in the last video, I'm going to skip through a bunch of things at the beginning of this video and go right to mitosis. So here we have cell division, and as you can see, remember I was talking about the G2 check and seeing if the DNA was damaged, and then I talked about the metaphase check point to make sure that all of the chromosomes are where they should be. So here we can see that this chromosome right here is lost. So in order for this cell to safely enter anaphase, he needs to get in line right here. So that's kind of the general idea behind that principle. Okay, so why do cells divide at all? Well, to make more cells, that's one reason, but also so that tissues can grow and tissues can replace the cells that they might lose. So that's called tissue renewal. And there's a specific kind of cell division that is specific to sexual reproduction, but you can also, if you are a single cell organism, just reproduce clonally by cell division as well. So if you used to be one cell, now you can be two. Another way to reproduce asexually is called budding. So this is a multicellular organism. I believe it is a coral polyp or something similar. It's an invertebrate regardless. I'm not exactly sure what it is. But regardless, the parent organism is going to grow a bud, and the bud is eventually going to reach the size and shape of the parent organism, and then you'll have two organisms that are genetically identical to each other. So that's how budding works. So budding either creates a multicellular new individual, or asexual reproduction in general just can include either budding, in which the child organism has multiple cells but they're all the same as the parents, or just a new cell that is the same as the old cell. All right, so this is a form of reproduction used by anything that's simple enough to do this, but when you get really, really complex, this stops being possible. So obviously humans, for example, we can't bud. I can't grow a new me out of my side, for example. So for unicellular organisms, this comes in the form of binary fission, which we already covered, so I'm going to kind of skip through this because we talked about this in the last video. So this is what I was trying to get to. So eukaryotes divide by mitosis, and this is because eukaryotes have a tremendous amount of DNA compared with prokaryotes. So they are basically needing to take extra steps to organize the DNA before it divides so that the DNA in the daughter cells is the correct amount. So as you can see from this picture, there are specific steps that are taken in order to achieve this, and you can also see that the parent cell looks identical to the cells that result from this division. So this is clonal reproduction of cells. So a cell is making another one of itself, and the two selves will be genetically identical to each other and to their parent. So in the last PowerPoint, I made sure to point out that mitosis is actually specific to the division of the nucleus, not to the entire cell. We have a different name for that. So it's nuclear division specifically by all complex cells except sperm and eggs, because these are gametes, aka not somatic cells. So this is necessary in order to renew tissues and to grow and to do all the good things that come with being made of multiple cells. Okay, so I explained this a little bit already, but we have a slide dedicated to it. So because we have so much DNA, it's really, really hard to divide it in half neatly if you don't organize it first. So it'd be like me handing you a wad of tangled rope and saying, hey, divide this in half exactly 50-50, go. That would be a huge pain in the butt, right? So instead, the DNA is packaged into chromosomes via supercoiling. So DNA is wrapped around histones, and the histones are threaded together into a chromatid, and a duplicated chromosome is only something that you see in a cell that's about to divide. So this karyotype that you're looking at here are the future daughter chromatids that are right before cell division. So the coiled structure of the chromosomes, remember you have histones, which are protein, and protein plus DNA, so a histone plus the DNA wrapped around it, that's called a nucleosome. And then the nucleosomes condense into chromatin, and the chromatin condenses into chromosomes. So this is a way to make sure that no segments of DNA are lost during mitosis. So here we have an example of the human genome. This is all 22 pairs of autosomes and the sex chromosomes, either XX or XY. So our total number of chromosomes is 46. And with these chromosomes, you get one from your mom and one from your dad. So students have a hard time thinking about the sort of specifics of chromosomes and karyotypes and what they mean. So let's say this chromosome here came from the individual's mom. This, and this, these are identical sister chromatids. Of a duplicated chromosome. And the same is true for the father's chromosome. So these are the same, but from dad instead. So you have 1, 2, 3, 4 chromatids, each containing the same genes, distributed across 2 duplicated and homologous chromosomes. So homologous means that there's the same genes on the chromosome from mom as there is on the chromosome from dad. So we've already seen and talked about this slide, so let's move right along. So recall that interphase is actually more complicated than just the cell sitting between divisions. So it's doing things like growing, doubling its DNA, and then growing and operating normally but with doubled DNA. And we talked about that in the last PowerPoint too, so I'm going to skip ahead here. So during interphase, this is what the DNA looks like. It's uncondensed and stringy. During S phase, the cell is busy copying all of its DNA, but the DNA at this point is still uncondensed, so the condensing happens later. So, unduplicated chromosomes look kind of like this, and duplicated chromosomes resemble an X. So these are the two identical sister chromatids. So chromosome duplication forms sister chromatids identical. And that's important, right? You don't want changes to the DNA. Those are called mutations. So during interphase, cell continues to operate normally in G2, but with duplicated chromosomes, and then you have the G2 checkpoint going on as well. But the meat of this PowerPoint is meant to be about mitosis. So let's talk about the mitotic phase. When a cell is ready to divide, it enters mitosis, and during mitosis, the nucleus dissolves, the DNA is divided in half, and the nucleus reforms around the resulting divided DNA. And this is carefully controlled through several stages, and we call these the phases of mitosis. So we have prophase, metaphase, anaphase, telophase, and cytokinesis. Hopefully, if everything goes according to plan, the result is that you have 2 genetically identical daughter cells, each with the correct amount of DNA, and each with all of the parts that the cell needs to survive. So the mitotic phase includes both mitosis, and cytokinesis. But it's important to remember that these two things are not the same. That's me trying to write a not equal sign sideways. It didn't work very well, my bad. So of course we're going to skip watching the mitosis video in the video, although that would be very meta, and go straight to mitosis. So during prophase, the nuclear membrane disappears, and the nucleolus disappears as well. So remember, nucleolus, this is the ribosome factory. So it's where ribosomes are made. But you don't need that when you're dividing. So the cell puts a pause button on making any more ribosomes or doing much protein synthesis. Excuse me. Because it's busy doing other stuff. So at this point, it's made all the proteins it needs to divide, and now it's busy dividing, and that's its primary job. The chromatin condenses into chromosomes. You get the separation of entrosomes, which are internal cell bodies, and the formation of the mitotic spindle. So you can see that there's little bits of endoplasmic reticulum that have now separated from each other because they're going to end up being the new endoplasmic reticulum of the new daughter cells. And you get the formation of the mitotic spindle, which is this structure up here. Sorry, I had to cough. Didn't want to cough into the mic. So, mitotic spindle is going to be this array of microtubules plus centrioles, and it'll take a more definitive shape in the next slide. So, this is a joke about condensation of DNA. So, dude, mitosis starts in 5 minutes. I can't believe you're not condensed yet. So that does take some time, which accounts for the length of prophase. Okay, so I promised a mitotic spindle that made sense and was easy to look at, and now we have one. So that's great. So this lemon-shaped array of microtubules that I'm bracketing here, this is the mitotic spindle. And what it does is imposes order and directionality on the inside of the cell so that there is a even division of DNA. So here we have duplicated chromosomes aligned on the metaphase plate. And two things are going to happen here. So we have the centrioles, which are anchoring the microtubules at each end, and we have two kinds of microtubules. One kind is attaching to the kinetochore at the center of the chromosome at a place called the centromere. So this is where the two chromatids connect. The other thing is that these two right here are going to polymerize towards each other when it's time for anaphase. So they're going to push against each other as the other kind, these kind, pull the chromatids apart. But for now, we're just making sure that all of the chromatids get to the metaphase plate. So we don't want any little lost chromatids or chromosomes at one or the other end of the cell. They've got to be all together. So now the nucleus is fully dissolved. This is classic metaphase. And you can see that the membrane-enclosed organelles are also kind of migrating to one or the other end. So they're not really hanging out in the middle. And that's important because they're going to be divided too, eventually. So the centromeres end up getting dissolved, which means that the sister chromatids are now no longer able to hold on to each other, and they end up being pulled towards opposite poles by these microtubules. Oops. There we go. Sorry. So these ones right here are doing the pulling. These ones right here are going to push against each other to move the two centrosomes further apart. And that creates this zone in the middle that is now ripe for cytokinesis. Okay, so once that has been achieved, the spindle disintegrates, so we get the depolymerization of microtubules, and you can see that the nucleolus and nuclear envelope are both reforming. So the chromosomes at this point begin to decondense because the membrane is forming around them, so they don't have to be condensed into neat little packages anymore. For now, division is over. Cytokinesis is going to initiate during this time. So telophase and cytokinesis, although they are separate things, they do overlap in time a little bit. So as telophase is proceeding, cytokinesis is initiating. So cytokinesis means cell cutting, and that's actually the breakdown of the word, so I don't have to do it for you. So this occurs during telophase, And plant cells also have to form a new cell wall. So we saw that in the last video that I made, where the Golgi vesicles align at where the metaphase plate used to be, and they end up helping to form the new cell wall. But in animal cells, it's a little simpler. You just get the contractile ring of microtubules forming the cleavage furrow, which you can see here. And, oopsie, sorry, I'm having problems today. This is what happens when you have a tired professor screencasting. So you can see the cleavage furrow right here, and that's going to continue contracting until the cell is pinched in half. So from one cell you get two cells, and we call these daughter cells. And both cells are exact copies of each other. And why is this important? Well, we've covered that a lot in your class. One particular reason that's compelling about why it's important is that if a cell is missing a chromosome, that means it's missing genes. And since we know that genes code for proteins, a cell that's missing genes is missing the ability to make a whole bunch of proteins. That is very risky for a cell. It doesn't produce cells that really survive very well. So the critical and ultimate outcome of mitosis is that you have two daughter cells that are genetically identical to each other. So this is how, even though you have lots of cell turnover in your body and you're not the same person cellularly that you were yesterday or 10 years ago, you look the same and your cells still have the same genetic information, even though they're constantly dividing and dying. So this is a joke about aging. You don't look anything like the long-haired skinny kid I married 25 years ago. I need a DNA sample to make sure it is still you. Just because our cells turn over doesn't mean that they're as good at their job when we're old. So when we get to some later points in the term, we'll talk a little bit about senescence and aging, but we're going to skip that for now. Okay, we've seen this slide. And this again is just the review slide that is mitosis and cytokinesis in overview. So this would be a good place to make flashcards from. So if you printed out this slide, for example, you could cut out the little pictures and put the bullet points on the back. So this last week in lab, I asked you to look at mitosis in onion root tips. So the apical meristem, which is the dividing region of an onion root cell, contains lots of cells undergoing, an onion root tip rather, contains lots and lots of cells that are busy undergoing mitosis. So you can see that here. We already covered that and these. So remember, when the cell cycle fails, so when controllers of the cell cycle are no longer in place, so tumor suppressor genes or proto-oncogenes, then you get cancer. And we talked a lot about that in the last video as well. And you'll investigate it a little bit in Lab 10 also. So checkpoints fail due to mutations. So in the simulation in the last PowerPoint screencast that I made, we saw mutations accumulating in a tissue, and that tissue eventually turning cancerous. So mutation of DNA, it's important to note, causes a mutation in the genetic instructions for making a protein. So what you get if you have damaged DNA is non-functional proteins. And if those proteins are critical for regulating the cell cycle, then you have a problem. So we talked a little bit in the last PowerPoint about viral oncogenes and the fact that viruses can turn off parts of the tumor suppression system in order for them to cause the cell to make more cells for the virus to divide in. And this is an example of this. So this right here is a wart. Yuck. So warts are caused by human papillomavirus, and human papillomavirus relies on skin cells. That's where it likes to be. So if it wants to live in skin cells, it's going to have your body make some extra skin cells and cover them over with keratin so they're nice and safe. So that's what a wart is. It's extra cells because the virus is wanting to grow and divide forever, so it's making your skin cells do the same thing. Okay, so here's the proto-oncogenes versus tumor suppressor genes slide again. Remember that you can either have a set of genes that pumps the brakes on cell division, so this is going to slow down the rate of cell division, or steps on the gas of cell division. So ordinarily, once more, these two sets of genes are active to control the rate of cell division in a particular kind of cell, but if one or the other of them gets damaged, you can have precancerous cells. And that, ladies and gentlemen, is the end of the mitosis PowerPoint. So I thank you for your attention, and I will see you for the next video.